Hello, my name is Dr. Blastocystis and I'm editor in molecular parasitology at the University of Kent in England. I'm the action chair of Blastocystis under One Health, which is an initiative to gather together all researchers working on blastocystis in Europe. I'm also the main organizer of the 42nd the National Blastocystis Conference that is taking place in um, Heraklion Crete in Aquila Hotel that we are here. Over the three next days, we are going to have talks on various aspects of blastocystis, and on day three, we're going to have a workshop discussing more on the epidemiology of blastocystis and what we need to do to expand this even further. Welcome to Crete.
I'm Hisao Yoshikawa from Japan. I'm working with the Blastocystis more than 30 years in my life. Now I retired last year and still ongoing with the research work on Blastocystis. So I also exciting in attending this Blastocystis conference in this time.
My name is Funda Doğrulmana. I am from Turkey, and it is good for me to be here because we will discuss many of blastocystic research.
I'm Eleni Gedekaki, and I am the vice chair of the Blastocystis under One Health Construction. I am here along with other delegates in a conference that is organized by the COST program and also the Microbiology Society. We're here to discuss and update ourselves on the current research on Blastocystis, a gut microorganism.
I'm Graham Clark from the London School of Hygiene and Tropical Medicine, and I'm here giving the plenary lecture, the first lecture of the conference on Blastocystis.
Hello, this is Varol Tol from Turkey. I'm a medical parasitologist, quite recently working with Blastocystis. I think Blastocystis is one of the most mysterious parasites there is. It's so common, yet we don't know so much about it.
Hello, my name is Constance Doyel and I'm a PhD student in the EcoFit team at the Pasteur Institute.
At various points, it was thought to be a yeast or a cyst of Trichomonas and a coccidian cyst. And it was a long time before we found out actually what it was.
A deeper dive into the lower abundance taxa that are associated with each subtype, because as I showed here, these are ones that had more than 20% abundance.
I was wondering, so you had xenic cultures of Blastocystis with other species, which were they the same to start with?
So what that tells us is basically that blastocystis has more adaptations in kind of anaerobic environment than Proteromonas has, and this is kind of obvious with what we do in the culture.
Question, do you intend to integrate the data, like try to integrate them at a different omics level?
Now we have, this amount of sample is 1,430 samples. That is a good number of samples, is a good data set.
Diet can, of course, have an impact on cardiometabolic health. So what we are seeing as a positive effect of blastocystis will be related to that.
Are they all existing at the same abundance, or is it dominated by one or two different species?
Hello, my name is Rune Stensgaard, and I'm from Denmark. I work in a diagnostic lab in a public health institute in Copenhagen, and I've been taking an interest in blastocystis for the past 20 years, I would say.
Hi, I'm Elisa Piperni, and I'm working on
computational metagenomics. We recently published a work on Blastocystis. Interestingly, we found typically avian subtypes ST7 and ST6 in 11 human samples, which is quite interesting.
The three histidine residue involved in the paper binding are mutated from the sub3 of Blastocystis.
Evidence linking some Blastocystis subtypes, particularly ST4 or ST7, with more, as has been identified as more pathogenic subtypes.
Some of the species that have other stage, like Negleria fowleri, which is the brain-eating amoeba in sweet water.
I thought maybe we should first arrange the evidence. There's like piling evidence about Blastocystis. We are all here researchers doing research of Blastocystis.
We don't have as much because we discussed various things. So the question is passenger or pathogen. We sort of ruminated that maybe it should be colonizer.
I mean, they will be paid for living normal lives, but as Truman people, realities. So when the children, for example, get diarrhea in their childhood times because of rotavirus, for example, they will all be, all data will be included in the database.
The process of doing this, or after just after you do it, quite fresh. It's like it takes a sample, or you put the sample in every time you are using it.
Monitor them to see how they respond, whether they develop symptoms or not, whether Blastocystis is retained, and what the Blastocystis load is.
For us, it's important also to consider also in the framework of One Health the identification of Blastocystis also in animals.
Subtype 3. We all know that subtype 3 is very diverse. And it may be that some of the differences between isolates of subtype 3 at its genome level will influence whether it can be cultivated. It is any clearer.
Next column, we can see the achieved percentage of the expected responses. The high prevalence is high responses belong to Turkey, and then Spain, and then France.
Our findings have expanded the number of Blastocystis subtypes recorded in free-roaming livestock in wild ungulates, while identifying subtypes at very low rates, which can help track down potential spillover events between wildlife and livestock species.
In total, we examined 23 animals from Zoo Košice, and we detected them by the flotation method to confirm the presence of other possible parasites.
Hi, I'm Pamela Koster. I'm working for Women for Africa Foundation. I'm collaborating with Carlos III Institution with David Carmena, and I made my PhD in different parasites, including Blastocystis.
Our study including 140 samples, distributed in human 50 samples and animals 50 samples, animal including 35 samples in sheep and 50 samples in goat.
We looked at Blastocystis and cryptosporidium in leafy vegetables destined for human consumption. So these were pre-washed and ready-to-eat vegetables. So the ones that you can get from the supermarket where they're treated beforehand and washed, placed in plastic bags. You can buy them and they're advertised to humans as ready-to-eat. You can put them on your plate and eat them straight away.
Arrows, several common bands were preferentially applied to the development of diagnostic primers, including humans, ethnic isolate NAND2 and B strain.
Yeah, it's correct. Carnivores, we don't see carnivores a lot with Blastocystis, and I don't think that any of the new subtypes or any new subtypes has ever been described in a carnivore for the first time.
There's genome sequencing and then there's genome sequencing. So in bacteria, if you sequence a genome, it's actually relatively straightforward to sequence bacterial genomes. They're mostly under 10 megabases. They don't generally have a lot of repeats.
I feel like I've benefited more from interacting with all of you who are speaking today than you've gained from me.
but you're both getting best a certificate from the Microbiology Society as well.
My name is Zuzana. I'm from Czech Republic, and our study is about the occurrence of blastocystis in gut-health individuals and their animals.
Hi everyone, my name is Beth Madrides. I'll just give you a quick presentation of my poster. So we looked at blastocystis Clostridium from in different supermarkets in Southeast England.
I'm Daisy and I'm from the University of Kent. This is my poster. We are working with the East Kent Hospitals University Foundation Trust to monitor the presence of blastocystis in patients before, after, and 6 months after. So we have full surgical removal for dental matters. Any doubt, you can just contact with us or other people that did this kind of mobility experience.
So that's why I just
But
wanted to
here
let you know.
there are more coming, there are different
But
opportunities.
someone could come to you.
Oh, we didn't compile the evidence so far. No one did this. So I believe that this will be a very high impact paper in the end.
Sample type is human, animal, and environmental sample. But as I mentioned before, the hardest one is human sample.
One, two, three. Over the last 3 days, we have a very eventful conference, the 4th International Blastocystis Conference, which involved talks from 43 participants, either oral or poster presentations. We had 2 sessions organized by the co-section on dissemination activities and future prospects on blastocystis, and on day 3 we had a workshop discussing how we can improve epidemiological studies. Overall, that has been a very successful conference, and we would like to thank, first of all, the Microbiology Society for supporting us, and secondly, the Blastocystis Under One Health Cost Action, funded by the European Union, that provided funding for lots of the participants to be here. Thank you, everyone, and hopefully I will see you at the 5th International Conference, wherever it will be. If you are wondering what is this, it's a giant microbe. Looks like a huge bubble, but this is actually blastocystis. Blastocystis is the most frequent eukaryotic microbe in our gut, but we have no idea what it's doing there. And this is the purpose of our studies and the whole blastocystis course action.